Good morning or good afternoon, everybody. Thanks to Eduardo, David, Nagomi, Peace, and all the organizers of the symposium for giving me such a great opportunity to present our recent work. Today, I'd like to talk about our recent finding about a non-membrane channel in dimerized calsequestrin in cardiomyocytes. As we know, the contraction of muscles is controlled by the transient increase of intracellular calcium. In cardiac muscle, calcium ions come to the cytosol via the calcium-induced calcium release mechanism, in which the voltage-gated hypocalcium channel in the cell membrane or T-tubules trigger the calcium release from the renoreceptors in the sarcoplasmic reticulum or SR. In the SR, calcium ions are stored by binding to calsequestrin because 1/3 of the residues of calsequestrin are negatively charged. With the difference of calcium concentration in the SR, there is a dynamic change in the molecular organization of calsequestrin in both skeletal and cardiac muscles. In skeletal myocytes, Dr. Rears and colleagues have shown that calsequestrin molecules are preferentially concentrated in the junction of SR, but with calcium depletion, calsequestrin mobility is much increased. In order to understand the molecular organization in cardiac muscle, we performed the cryo-electron tomography of cardiomyocytes. After preparation of cryo-slice samples, we rotated the sample every 2 degrees to take transmission electron images, and then these images are used for 3-dimensional reconstruction. In the reconstructed image, we can identify T-tubules and the junction of SR. Between the T-tubule membrane and the SR membrane, we can see electron-dense renoreceptors. Also, there are electron-dense signals inside the SR, which can be recognized by CSQ antibodies in separate immunoelectron microscopic experiments. Here is an example of 3-dimensional rendering of a calcium release unit, where renoreceptors are colored in red, and the CSQ2 network is colored in green. The thickness of the CSQ signals is around 6 nanometers, which is comparable with the size of a calsequestrin-2 molecule or its dimer. By docking calsequestrin-2 dimers onto the reconstructed CSQ2 network, we can see that the network is composed of a single layer of calsequestrin-2 dimers. It is well known that many mutations of CSQ2 induce catecholaminergic polymorphic ventricular tachycardia or CPVT. Recent reports have shown that some of the CPVT-inducing CSQ2 mutations exhibit dominant genetics. It is hypothesized that calcium may flow among CSQ2 molecules such that a mixture of wild-type and mutant molecules will block the conductivity of the whole network. In order to test this intriguing possibility, we crystallized the human CSQ2 protein in a solution that mimics the high calcium concentration in the SR and determined the crystal structure of calcium-bound human CSQ2. In the monomer of CSQ2, there are 3 thyrodoxin-like domains which are shared in common with all CSQ structures. All the CSQ2 molecules existed in dimers in the crystal. The unique feature of our structure is that we identified 36 metal ions assigned as calcium ions based on the orbit density map and the condition of crystallization. These calcium ions are distributed on both sides of a CSQ2 monomer, binding to negatively charged amino acid residues, which are marked in red. Notably, on the interface side of a monomer, the negatively charged residues are aligned to form an arc-shaped groove hosting a line of 15 calcium ions. In the cut open view of a CSQ2 dimer, this negatively charged groove appeared as a channel-like structure across a CSQ2 dimer. From the side view of a calcium-bound CSQ2 dimer, the channel-like structure was formed when two monomers bind in parallel with each other. In contrast, in previously reported CSQ2 dimer without calcium binding, the two monomers are bound with an angle of 35 degrees. Here is a movie showing calcium-dependent change of a CSQ2 dimer. In the absence of calcium, the negative charges from both monomers repulse each other, keeping a hollow inter-monomer space. In the presence of millimolar calcium, by contrast, calcium ions attract negative charge from both sides, holding monomers together to form a channel-like structure. Within the putative channel, calcium ions are hosted by negatively charged residues. This is a summary of calcium positioning collected from 14 monomers. We can see that both calcium ions and residue side chains exhibit dynamic flexibility. We speculated that these calcium ions may move among the adjacent residues, similar to the movement of potassium ions through the pore of a potassium channel. To explore this intriguing possibility, we analyzed the geometry of putative channel using the whole software, which is commonly used to map the geometry of ion channels. The analysis showed that the channel has two symmetrical arms with a minimum radius of 1.7 angstrom. This radius is wider than the ionic filter of a typical transmembrane calcium channel CAV 1.1, whose bottleneck radius is around 1.4 angstrom. This suggested that the CSQ2 channel may be permeable to partially dehydrolyzed calcium ions. Now, the challenging question is how to test the conductance of a non-membrane channel. Without a membrane to host the channel proton, patch clamp cannot apply. In collaboration with Professor Xuefeng Guo in the College of Chemistry of Peking University, we designed a graphene-based nanoelectronic technology to measure the conductance of a CSQ2 channel. We prepared pairs of graphene nanoelectrodes with a 6 to 7 nanometer gap. Then we functionalized the graphene electrodes, leaving a free thioreactive end group, the red one. To link CSQ2 molecules to the thioreactive electrodes, we introduced the assisting labeling site at the residue 348. In this design, each CSQ2 monomer in the dimer can be connected to an electrode by the formation of a disulfide bond. The connection of a CSQ2 dimer to the nanoelectrodes can be visualized by atom force microscopy. To measure the electrical property of a CSQ2 dimer, we applied different voltages. In the absence of a CSQ2 dimer, a voltage of 50 mV induces a Farad current less than 2 picoampere. In contrast, in the presence of a CSQ2 dimer, the current was increased by more than 1 order of magnitude, showing a conductance more than 600 picosiemens. When the disulfide bonds was reduced by DTT, the conductance returned to the buffer level, confirming that the large conductance was attributable to the conduction of CSQ2 dimer. When the calcium concentration in the buffer was reduced from 1 millimolar to 100 nanomolar, the conductance was also decreased to the buffer level. Replacing calcium with strontium also compromised the conductance. These results supported calcium as the charge carrier through the channel. Because negatively charged residues are distributed both on the surface and in the interface of a CSQ2 dimer, to determine whether the conductance was generated through the channel or bypassing the channel, we replaced the negatively charged aspartate at the 309 site of the bottleneck region by a non-charged asparagine. Atom force imaging and other experiments showed that the mutation did not influence the dimerization of CSQ2. Nanoelectronic measurements showed that the 309 replacement suppressed the conductance by 76%, indicating that at least 3/4 of the total conductance was carried through the channel. Most of the above tests are chemical or physical experiments to test whether the CSQ2 channel is physiologically sound in intact cardiomyocytes. We constructed adenovirus to overexpress Y-type and 309 mutant CSQ2 in normal ventricular cardiomyocytes. In the Y-type group, electrical pacing evoked calcium transient with clear upstroke and clean diastolic background in the presence of isoproterenol to stimulate beta-adrenergic receptors. In contrast, in the 309 mutant group under the same condition, the calcium transients were tangled with chaotic calcium waves and delayed after depolarizations, or DADs. When the development of a DAD reached the threshold of sodium channel opening, a spontaneous action potential or triggered activity was initiated. Chaotic calcium waves, DADs, and triggered activities are cellular hallmarks of CPVT, suggesting that blockage of CSQ2 conductance underlies the pathogenesis of CPVT. Of note, the 309 mutant CSQ2 was expressed in addition to the natively expressed Y-type CSQ2 in normal cardiomyocytes. The defective intracellular handling in 309 mutant group was not attributable to the calcium buffering capacity of CSQ2. Rather, this phenomenon was consistent with the scenario that CSQ2 molecular network may facilitate the calcium flow in the SR. Indeed, in our structure analysis, we observed that the CSQ2 channels are interconnected. The interconnection of CSQ2 channels explained why a mixture of Y-type and mutant CSQ2 channels paralyzed the CSQ2 function, and why some of the CSQ2 mutations exhibited dominant genetics in causing CPVT. Here is a summary of the CPVT-inducing mutations. The yellow ones are known to occur in the hydrophobic core and may induce misfolding of the molecule. Notably, the green ones are mutations around the 309 bottleneck region. We think that these mutations may influence the channel conductance directly or indirectly. The cyan ones, although distributed widely, they are all involved in the interconnection of CSQ2 channels in our calcium-bound CSQ2 structure. Therefore, our findings of the CSQ2 non-membrane channels provide a novel mechanism for the calcium handling in the cardiac SR. Each CSQ2 dimer formed a calcium conducting channel. The CSQ2 channels are interconnected to form a channel network, which provide a nanohighway for fast diffusion and equilibrium of calcium ions in the SR. Otherwise, the diffusion of calcium ions in the heavy buffer environment would be extremely slow, which may cause inhomogeneity in local SR calcium concentration. Mutations that block or interrupt the calcium highway will lead to heart disease such as CPVT. This work was done by postdoc fellows in our lab, Drs. Shi-Qiang Wang, Lin Li, and Li-Peng Wang, and PhD students Xinlei Ma and Hongtao Li, in collaboration with Professors Xuefeng Guo and Chen Song. Zhihao Liu in Guo's lab performed the nanoelectronic measurements, and Dr. Hua Yu in Song's lab performed the computational analysis of the non-membrane channel. Thank you for attention.